Instrumental insemination is an essential tool for both research and stock improvement. Our increasing knowledge of bee genetics and the advances in molecular techniques offer new opportunities in bee breeding. Improvements in equipment design provide high repeatability and personal choice in techniques. With proper treatment and care, instrumentally inseminated queens perform as well as naturally mated queens. The insemination procedure is presented here in step-by-step detail with troubleshooting sections to help you avoid common mistakes. My goal is to help you utilize instrumental insemination and take advantage of the potential this offers. In nature, honeybee queens mate with numerous drones from diverse genetic backgrounds. Instrumental insemination provides the ability to control mating, enabling the study of bee genetics and the development and maintenance of selected stocks. With the sequencing of the honeybee genome comes exciting new opportunities. We are beginning to identify the function of specific genes and may learn how to turn these genes off and on to control diseases and parasites. To ensure success with instrumental insemination, every step of the procedure, from the rearing of queens and drones to the establishment of laying queens in colonies, must be given proper attention. The first step in the process is to provide optimal queen and drone rearing conditions. This requires strong, healthy, well-fed colonies that are free of pests and disease and chemical treatments and residues. Drone production is just as important as queen rearing. Drones, which are seasonally produced, have a high rate of attrition and are sensitive to stress. Obtaining a plentiful supply of mature drones is often the limiting factor in an insemination program. To save labor, virgin queens and drones can be banked in nursery colonies, but keep in mind that the queen can be subject to injury from aggressive worker bees. As a result, the practice of banking should be minimized as much as possible. For banking, use queenless nursery colonies that are well fed and supplied with young, open brood. Here you see a cloak divider board used to create a queenless nursery in the top of this colony. For best results, use quality rearing conditions, establish each virgin queen in a nucleus colony. Queens can be caged as they are extremely active and will take flight. The optimal age for virgin queens for insemination is 5 to 10 days post-emergence, though this may vary with the season and stock. Drones, which are slower to develop, should be at least 2 weeks of age or older. And remember, try to limit banking practices as much as possible. In 1927, Dr. Lloyd Watson demonstrated the first successful insemination technique using these tools, which he made himself. This device uses a hand-blown glass syringe and a wooden bed to which the queen is tied with a silk thread. A pair of forceps is used to lift the sting. The Mackinson instrument, in use from the late 40s through the 80s, is the first prototype from which most modern instruments are modeled. Its simple movements are made by sliding the hooks and syringe. Today's instrument designs use micro-manipulators which offer more precision and provide high repeatability. The German-made Schley instrument makes use of micro-manipulated syringe and ball and socket joints for precision control. The post can be moved along the base and the syringe can swing forward and backward, offering a wide range of precision movements. A variety of sting hooks and forceps can be used. The Schley Compact 2 has a shortened base to accommodate a wider range of microscopes and can also be accessorized for the left-hand user. The pressure-equipped forceps are featured here, and the instrument is updated with a Harbaugh syringe. The Harbaugh large-capacity syringe was designed for semen storage. The easily detachable capillary tubes and tips provided unlimited capacity and an easy means to store and ship semen. The micrometer, housed in a separate stand, is used to accurately measure semen volume. The Sventi instrument, made in Denmark, is designed on a set vertical plane. It uses a joystick type of micro-manipulator for the hooks and syringe, reducing hand movements. European instruments, such as the Schley and Sventi featured here, use a standard plunger-type syringe with a capacity of 50 microliters. The simplified Kunert-Laidlaw instrument is designed to be economical and easy to construct. This device uses a pair of handheld forceps, replacing the sting hook. Modeled after the Kunert-Laidlaw instrument, the Latchel instrument makes use of a micro-manipulated syringe. Made in the Czech Republic, the Weasley instrument is a hybrid of the Mackinson and German models and uses a large capacity syringe. There are a variety of other manufactured instruments as well as many other homemade versions like this gold-plated instrument made by jeweler Mark Jones. When choosing equipment, consider these points: precision and repeatability, a wide range of adjustment, the choice of syringe design, and the type of hook or forceps used. The first step is to select drones for semen collection. A flight box provides easy access to drones. Drones have a high rate of attrition, and losses can occur during rearing, maturing, and banking. Therefore, rear a plentiful supply of healthy drones. The first step is the partial aversion. At this stage, the abdomen contracts and the cornu are exposed. These are horn-like structures seen here. The second step is the full aversion. Here you see the endophallus is fully averted and the semen is exposed. The exposed semen rests on a bed of mucus and is easily distinguishable. The semen is a marbled creamy color compared to the white mucus layer. To obtain the partial aversion, grasp the drone by the head and thorax. Crush the thorax, holding the drone dorsally and ventrally. During partial aversion, the abdominal muscles will contract, causing the abdomen to feel hard to the touch. During this stage, pressure should not be placed on the anterior or sides of the abdomen, and care must be taken not to crush the abdomen. To obtain the full aversion, apply pressure along the sides of the abdomen with the thumb and forefinger, working from the base toward the tip of the abdomen. Roll your fingers forward in a strong and steady motion. This will avert the endophallus and expose the semen. If you are right-handed, obtain the partial aversion using the right hand, then transfer the drone to the left hand before completing the full aversion. This will leave the right hand free to manipulate the syringe for semen collection. During this process, hold the drone downward to prevent the endophallus from flipping back onto your fingers. This will cause contamination. During the aversion process, drones often defecate, especially when banked. The feces is very acidic and may cause skin and eye irritation. Take care to hold the drones away from your face and away from the insemination equipment during the aversion process. The semen is a nutrient-rich medium for bacterial growth. Care must be taken not to contaminate the semen, hooks, or syringe tip. Here is a test to determine drone maturity. If the abdomen does not contract, the drone is immature. Notice a lack of color in the cornu. These are clear, indicating the drone is immature. Discard and pick another drone. Notice on this drone the orange-yellow color of the cornu, indicating the drone is mature. If the drone is too mature, it may pop when handled. Remember these points. For the partial aversion, grasp the head and thorax. Crush the thorax to stimulate the aversion. Avoid pressure on the abdomen and test the drone for maturity. This will save time later. For the full aversion, apply pressure from the base to the tip of the abdomen. Take care to avoid contamination, and then position the endophallus under the microscope for semen collection. To prepare for semen collection, first make sure the syringe is properly prepared. Draw a small airspace into the tip of the syringe and make sure the plunger is drawn forward to allow sufficient space for semen collection. Collect approximately half a microliter of saline in the taper of the tip. Bring the drone's endophallus and the syringe tip into focus. Expel a small drop of saline into the semen of the endophallus while maintaining the airspace. Skim the semen off the mucus layer, taking care to avoid collection of mucus. After semen is collected from the endophallus, take up another small airspace and a drop of saline to prevent drying of the semen. Avert the next drone. Expel the drop of saline and the airspace in the tip onto the semen of the next drone. Bring the tip into contact with the semen, again being careful to avoid the mucus layer. Use the tip to skim the semen off the mucus layer. Do not collect any air bubbles with the semen. Repeat this procedure until the desired amount of semen is obtained. If you accidentally collect mucus during the semen collection process, use the following procedure to tease it out. Collect and expel a few microliters of saline in the tip. Work it back and forth to loosen the mucus in the tip. Once loose, expel the mucus with the extra saline. A mucus plug in the tip will cause the syringe to be unresponsive. Do not allow buildup of excess pressure in the syringe. If the mucus plug is lodged in the taper of the tip may have to be removed and back flushed. With experience, this can be avoided. Collect a drop of saline to make contact with the semen on the endophallus. Be sure to avoid the mucus layer. Skim the semen off the endophallus and avoid collection of bubbles and extra saline. Once the semen extraction is complete, the column of semen should be uniform without air bubbles or light areas. Light areas indicate the use of too much saline. To position the queen in the holding tube, first place the queen head first into the backup tube. Hold the backup tube flush against the queen tube. The queen should walk into the tapered holding tube. Place this over the carbon dioxide valve and properly position the queen. When properly positioned, the terminal segments of the queen's abdomen will protrude from the tapered end of the queen tube. If the tube is not tapered, hold the queen into position until she becomes motionless. Carbon dioxide is used to anesthetize the queen during the insemination procedure. Next, position the queen at a 45-degree angle so the dorsal side of the abdomen is to the right and the ventral side to the left. The hooks should be positioned above the queen holder, level with the tip of the queen's abdomen. There are a variety of hooks and forceps available to manipulate the sting. All are designed to lift the entire sting structure. The ventral hook with its right-angled bend is designed to stabilize the ventral side of the abdomen during the insemination procedure. The Schleif forceps are mounted on a handle. The forceps are activated by a push-button control. For best results, use light pressure to grasp the sting toward the tip. The simplified instruments using the handheld forceps, the ventral hook is placed into position first. The sting is then grasped and lifted with the forceps. Another choice is the perforated sting hook. This tool is designed with a hole to thread the sting. To use, place the hook above the sting and thread the sting through the hole. Take the hook to the base of the sting and lift the sting. As the sting is lifted, the bend of the sting will secure this from slipping. Another option is the ruby jewel perforated hook. The advantage is the transparency of the ruby. The bevel on the underside of the jewel guides the sting into the hole. The jewel hook is used in the same manner as the perforated hook. Once the sting is threaded, the bend will secure the hook while lifting. The classic sting hook has a flattened and flared spoon shape, which is used to lift the sting. This has largely been replaced by the newer hook designs. For ease of manipulation, place your fingers close to the hooks on the inside of the holding post. Your hand should rest comfortably and securely so they are steady. To open the queen, use both hooks simultaneously. If you have difficulty opening the queen, the ventral hook may be inserted first, followed by the sting hook. The ventral hook can be turned sideways and used to stabilize the sting. When the sting is threaded, move the ventral hook back into position and lift the sting with the perforated hook. It is important to have proper alignment of the hooks when opening the sting chamber. Make sure to follow the 45-degree alignment of the queen holder and syringe. If you have difficulty, check the alignment and reposition the hooks. A common mistake is to push the sting downward, placing pressure on the poison sac and distorting the alignment. Another common mistake is to pull the ventral hook to the left. This will cause misalignment, making the insemination difficult. When positioning the queen in the instrument, note the state of the rectum. If distended, remove the queen and place her on a paper towel to defecate. Another queen may be inseminated during the wait. Consider these points. Position the queen, use the ventral hook to stabilize the abdomen, grasp and lift the sting, and follow proper alignment. In this side view of the queen's abdomen, notice the position of the valve fold, oviducts, and spermatheca. The valve fold is an invaginated tissue that blocks passage of semen into the oviducts. This must be bypassed to properly inseminate the queen. To inseminate the queen, the syringe tip is used to lift the valve fold, enabling delivery of semen directly into the median oviduct. The valve fold is not readily visible. When the hooks are properly positioned, you can see the V of tissue that defines the location of the valve fold. The valve fold is located beneath the V. Locate the V of tissue, position the tip above and dorsal of the apex of the V. Insert the tip approximately 1 millimeter, Then move the tip slightly ventrally, increasing the incline of the tip in order to bypass the valve fold. Insert the tip another 0.5 to 1 millimeter into the median oviduct, lifting the valve fold ventrally. A slight zigzag motion of the tip is used during the procedure. If the tissue moves with the valve fold, it has not been bypassed. Here, the tip is resting on the top of the valve fold and will cause the semen to leak. Remove the tip and try again. Insert the tip close to the sting hook, to the right of the V, and use a zigzag motion to bypass the valve fold. Consider these points. Locate the V that defines the valve fold. Insert the tip approximately 1 millimeter and lift the valve fold ventrally. Insert the tip another 0.5 to 1 millimeter and test the positioning of the tip by delivering a drop of saline. Then deliver 8 to 10 microliters of semen. Once the tip is properly positioned, eject a small drop of saline along with a small airspace in the taper of the tip. This allows you to test if the valve fold has been bypassed. If there is no backup, proceed to inject the semen. Deliver a measured amount of semen. 8 to 10 microliters is the standard dose per queen. Withdraw the tip. The tip should be removed cleanly with as little semen leakage as possible. If semen backs up, as in this example, the valve fold has not been bypassed. Note that the tissue moved with the syringe. Reposition the tip and try again. Proceed each insemination with 1 microliter of saline to test if the syringe has been properly positioned. Semen leakage can also occur if the tip has not been inserted far enough or if the syringe has pressure buildup. If you continue to have difficulty with the insemination, check the position of the hooks and alignment of your instrument. Are the hooks pulled too far to one side? Is the sting hook pushed down instead of lifted up? Does the queen need to defecate? After insemination, collect a small airspace and a drop of saline to proceed the next insemination. This prevents the drying of semen and acts as a lubricant for the next insemination. While the queen is still anesthetized, mark and clip the queen. When queens recover from the effects of carbon dioxide, they are often hungry. The carbon dioxide treatment may cause regurgitation. After insemination, queens should be immediately returned to their colonies. During the next 48 hours, the sperm will migrate from the oviducts into the queen's spermatheca. During this critical time, care of queens is extremely important. The conditions will influence the number of sperm stored. While learning the technique of instrumental insemination, a good test for checking your success is to observe the spermatheca. Color and density will give you a good estimate of how much sperm has migrated into the spermatheca. Here we see the spermatheca of a virgin queen, which is clear and transparent. This is the spermatheca of a mated queen. Notice the marbled creamy color. If the spermatheca contains only a small amount of sperm, the appearance of the spermatheca is cloudy, as seen here. To dissect the spermatheca, first, crush the thorax of the queen, then grasp the last segment with a pair of forceps or fingernails and pull. Embedded in the tissues of this terminal segment will be a round, white sphere with a diameter of about 1 millimeter. This is the spermatheca. The spermatheca will initially appear whitish and rough. This is because a network of trachea covers this. The tracheal net is removed by rolling the spermatheca gently between the fingers. As you can see here, the queen has been successfully inseminated. Semen can be held overnight or for several weeks at room temperature in sealed capillary tubes. Avoid temperature extremes and do not refrigerate semen. This will cause sperm mortality. Storage tubes can be sealed either with petroleum jelly or a piece of silicone tubing and a glass bead as seen here. Draw the semen completely into the capillary tube, remove the tip, then disconnect the semen-filled capillary tube from the syringe. Place a petroleum jelly plug into one end of the tube with a sterilized spatula. No airspace is needed. The tube can be cut to size if necessary. Be sure to allow space for the petrolatum plug at the other end. To place the second plug, reconnect the tube and move the column of semen flush with the other end of the tube. Detach the capillary tube and place the petrolatum plug. The semen is now ready for shipment. Again, take extra care to avoid contamination of the semen. When preparing for insemination, make sure to properly sterilize and prepare the syringe in advance. Attach the tube to the syringe, leaving the petrolatum in place. This acts as a separator. No airspace is needed. Remove the petrolatum plug on the tip end of the tube before attaching the tip. Be sure to sterilize and rinse the tip with saline solution. Next, collect an airspace and some fresh saline. Now you're ready to proceed with the insemination. The optimal condition is to directly release the queens into nucleus colonies. Queens should be introduced as queen cells or virgin queens, and for convenience can be caged. To facilitate acceptance, spray the queen and bees with a fine mist of sugar water. Be sure to place an excluder over the entrance until egg laying begins. Another option, when direct release is not possible, is to use a push-in cage. Place the cage over emerging brood so young bees will emerge to attend the queen. A review of studies from 1946 to the present comparing instrumentally inseminated queens and naturally mated queens clearly demonstrates the competitive performance of instrumentally inseminated queens and the importance of proper care. Comparison studies measured brood and honey production and queen longevity. The results are categorized into 3 groups. Group 1 includes 5 studies showing equal performance of instrumentally inseminated and naturally mated queens. Group 2 includes 6 studies showing higher performance of instrumentally inseminated queens, which is attributed to the ability to control mating. Group 3 includes 1 study showing higher performance of naturally mated queens. A review of factors affecting queen performance conclusively shows that the treatment of queens has a significant influence. The Group 3 findings can be attributed to the different treatment of queens. Instrumentally inseminated queens in Group 1 and 2 were inseminated at an age range of 5 to 12 days and were given semen dosages ranging from 8 to 12 microliters. These queens were introduced into nucleus colonies or package bees with minimal or no banking. In the Group 3 study, instrumentally inseminated queens were inseminated while 2 or 3 weeks old and confined to cages and bank colonies for another 2 or 3 weeks before introduction. Queens were given 2 small semen doses of 2.7 microliters. The queens were caged and shipped at the start of egg laying, then introduced into large colonies or package bees. The low sperm counts and lower rates of production survival of the instrumentally inseminated queens in Group 3 can be attributed to the methodology used. Queens inseminated past their prime receptive mating age store significantly less sperm. Confinement to cages after insemination also reduces the efficiency of sperm storage and often subjects queens to injury by aggressive worker bees. Queens undergo dramatic physiological changes in preparation for egg laying. Many factors influence the rate of these changes and affect performance. Queens that are inseminated at a young age, allow free movement, and are well attended by worker bees mate with more drones and store more sperm. Gaining proficiency in the technique of instrumental insemination requires practice and attention to detail. The first attempts may seem awkward and frustrating. Concentrate your efforts on mastering each step of the procedure. Success and efficiency will develop with experience. To achieve this requires proper insemination techniques, beekeeping skills, and proper pre- and post-insemination care of queens. Instrumentally inseminated queens are capable of heading productive, full-size colonies. For effective evaluation of breeding stock, it is essential to allow these queens to build strong colonies. Instrumental insemination also enables the creation of specific crosses that do not occur naturally, providing significant advantage to research and stock improvement. For example, a single drone can be mated to 1 or several queens, isolating or amplifying a specific trait. Varying degrees of inbreeding can be created to produce different relationships within a colony. The ability to pull and homogenize semen from hundreds of drones and inseminate a portion of this to 1 queen enables unique mating system designs and simplifies stock maintenance. Another advantage of instrumental insemination is the ability to store and ship semen. The shipment of semen, rather than live bees, minimizes the risk of spreading pests and diseases. Instrumental insemination is a valuable and essential tool for honeybee research and stock improvement. With the increasing challenge of pests and disease, selective breeding offers the best long-term solution. Sophistication in instrument designs makes the technique easier to learn and highly repeatable. Add to this our increasing knowledge of bee genetics, breeding system designs, and selection methods. Instrumental insemination truly offers an exciting future potential.